Welcome to the pod Biomanufacturing and Fermentation Technology. In this episode, we translate microbial science, process engineering, and manufacturing realities into insights that scale.
Usually when we talk about manufacturing, or a mechanical process, there is this underlying expectation of absolute visible precision.
Right, like engineering a car engine.
Exactly. You put a camera on the assembly line, you watch the robotic arm weld a door, and you can just point and say, there it is. The door is attached.
It's binary. It's clean.
Yeah, fastened or not fastened. But step into the world of modern biomanufacturing, into a massive commercial fermenter, and suddenly that visibility just vanishes entirely.
It really does.
We're looking at a manufacturing landscape that is, honestly, a murky, bubbling soup of microscopic cells. And decoding that black box, figuring out how modern biomanufacturing actually sees the biology in real time, is exactly what we are unpacking today.
It's a massive challenge.
It is. And welcome to the deep dive, by the way. Our mission today is to decode the digital brain of modern fermentation. We're looking at a stack of technical sources breaking down how bioprocessors operate, specifically comparing SCADA systems, PADA, and the complex data management layers, orchestrating the whole thing.
And, the evolution of these systems dictates how much of the actual biology we can see, understand, and control in real time.
Right.
I mean, you have billions of living organisms in a steel tank, and they don't exactly come with a digital dashboard showing you their metabolic rate.
No, they definitely don't.
For a long time, the way we monitored them was incredibly rudimentary, essentially relying on simple digital chart recorders just to track basic physical stats.
And that matters to you, the listener, because whether you are prepping for a high-stakes biotech scale-up meeting or trying to understand how modern life science factories actually produce the medicine you take, or, just love nerding out on the bleeding edge of automation.
Which is always fun.
Yeah. This deep dive is your shortcut to being well-informed without the overwhelming jargon. So, okay, let's unpack this.
Let's do it.
Everyone knows SCADA is the operational bedrock of any plant. It handles the dissolved oxygen, the pH, the temperature.
Physical environment.
Right. But standard SQL databases completely choke on the volume of data a fermenter produces. That is why our sources heavily emphasize continuous or batch historians.
Like the Delta V continuous historian or AV-EVA historian.
Exactly. We aren't just saving a spreadsheet to a desktop here. We are dealing with high-frequency time series data.
Well, think about the sheer velocity of information hitting the system. You might be pulling readings from hundreds of sensors multiple times a second, for weeks on end during a long mammalian cell culture batch.
Wow.
And a standard relational database is designed to link disparate pieces of information, like matching a customer name to an order number.
Right, it's transactional.
Exactly. It's not built to ingest a fire hose of raw physical states, events, and alarms without missing a millisecond.
Yeah.
Process historians are highly optimized to store massive, continuous streams of numerical arrays very efficiently.
And in the regulated life science world, missing a millisecond is a massive legal problem.
Oh, absolutely.
If you're manufacturing a biologic drug under 21 CFR Part 11 in the U.S. or Annex 11 in Europe, regulators demand absolute, continuous proof that the batch was made safely.
They don't mess around with that.
No. So properly configured SCADA systems are maintaining unalterable audit trails. Like if an operator manually changes the set point for the agitation speed, how fast the paddles are stirring the broth at 3:00 a.m., the historian records exactly who did it, when, and what the previous value was.
Because the regulatory framework demands that level of granularity.
Right.
If an adverse event happens with a patient months down the line, the pharmaceutical company must be able to pull up the electronic batch record and prove that the fermenter's temperature never deviated beyond the specified parameters, even for a few seconds.
That's intense.
It is. SCADA provides that trusted, encrypted operational shell. It physically connects to the programmable logic controllers, the PLCs on the equipment, and executes the control logic. If a valve needs to open to release pressure, SCADA acts.
So if SCADA is the operational shell, I see it kind of as the autonomic nervous system of the human body.
Oh, I like that.
Yeah, like your autonomic nervous system keeps your heart beating. That is the agitation in the tank. It makes you sweat to keep your body temperature stable.
A reflex.
Exactly. It reacts to immediate physical stimuli to keep the organism alive without you having to consciously think about it. SCADA is reacting to the physical environment, keeping the body of the fermenter stable.
That analogy perfectly captures the baseline function, and if we connect this to the bigger picture, it highlights how the system is evolving.
How so?
Well, modern bioprocess platforms now use standardized interfaces like OPC UA and web services to expose open APIs. SCADA is no longer a standalone isolated island in the plant.
Right, it's talking to other systems.
Yes,
Yeah.
vendors are building advanced dashboards and KPIs right on top of the historian, so process teams can visualize the data and compare batches in real time. It functions as a fully integrated node in a much larger digital manufacturing stack.
But there's a massive limitation built into that autonomic nervous system.
What's
As amazing as SCADA is at maintaining the physical environment,
Yeah.
keeping the room at an exact temperature with the perfect amount of oxygen, it's still just managing the room.
Right.
It cannot tell you what the cells are actually doing inside that room. It knows the pH, sure, but it doesn't know if the cells are multiplying rapidly, if they're starving, or if they are actively producing the target protein.
That is the blind spot. To bridge that gap, you have to move beyond physical parameters. You have to measure and control critical quality attributes, or CQAs.
And the CPPs, critical process parameters.
Exactly. This requires a completely different conceptual framework from SCADA.
Which brings us to PAT, Process Analytical Technology. We already established you can't just stick a standard probe into a tank and get a reading for biology.
No, you need advanced spectroscopic sensors.
Specifically in situ or at-line, near-infrared, NIR, and Raman spectroscopy.
Yeah.
You are literally shining specialized lasers or infrared light directly into the broth to interact with the molecules.
The physical mechanism there is just fascinating. When you shine a laser into the broth using Raman spectroscopy, most of the light scatters normally, but a tiny fraction of those photons actually interact with the chemical bonds of the molecules in the soup.
Like the glucose, the amino acids, the proteins.
Yes. When they hit those bonds, they bounce back with a slightly different energy level. This inelastic scattering creates a distinct spectrum. It's effectively a high-dimensional chemical fingerprint of everything in the tank at that exact second.
Wow. But a complex light scatter doesn't just print out a label saying, 10 grams of glucose.
No, it definitely doesn't.
It has to be translated. And that is where soft sensors come in. We are talking about multivariate or chemometric models, specifically algorithms like PLS, partial least squares, or PCA.
Principal component analysis.
Right. They are mathematically trained to take that massive array of light data and convert it into a real-time estimated value for biomass or product concentration. It's software creating a virtual probe where a physical probe simply can't exist.
And the mathematics behind PLS is what makes this translation possible. A raw Raman spectrum might contain thousands of individual data points for every single reading.
Thousands.
Oh, yeah. And you cannot map thousands of variables to a single glucose concentration using basic algebra.
Right.
PLS reduces the dimensionality of that data. It finds the underlying latent structures, the specific patterns in the light scatter that consistently correlate with changes in glucose or biomass while filtering out the noise.
Okay, I see.
Once you isolate those correlations, your virtual probe becomes incredibly accurate.
And once you have that real-time biological data, you unlock advanced controllers. Instead of a reactive PID loop that just waits for oxygen to drop before turning up the air, PAT enables model predictive control, or MPC.
A huge leap forward.
Yeah, MPC acts more like a grandmaster chess player. It uses the soft sensor data to simulate multiple future trajectories.
It looks ahead.
Exactly. It predicts that the cells will hit an exponential growth phase and need a massive spike in oxygen in 20 minutes, so it starts adjusting the feed profiles proactively right now.
The shift from reactive control to predictive control fundamentally changes batch yields. You are no longer stressing the organism by waiting for it to starve before feeding it. You're anticipating its metabolic needs based on actual real-time biological feedback.
I have to push back on the architecture here for a second, though.
Okay, go ahead.
Wait, if SCADA is already collecting massive amounts of high-frequency data from the fermenter-like temperature, off-gas, pH, why do we need this entirely separate PAT layer to do the math?
That's a fair question.
I mean, can't the SCADA system just ingest the Raman data, run the PLS algorithm itself, and guess the biomass?
Well, the distinction comes down to computational architecture and the fundamental nature of the data.
Okay.
SCADA is built to process basic single-point tags at lightning speed. A temperature probe gives you one number, 37 degrees. SCADA logs that and moves on.
Super.
Right. But a Raman spectrometer is feeding you a complex, multidimensional matrix of thousands of data points every few minutes. SCADA simply isn't designed to perform heavy, iterative matrix algebra on the fly while simultaneously ensuring the tank doesn't overpressurize.
Oh, I get it. It's the difference between a reflex and a deep analytical thought.
Yeah.
SCADA needs to keep the reflexes firing without interruption.
Precisely why PAT sits on top of or alongside SCADA. PAT ingests the basic SCADA tags as contextual inputs, combines them with its own spectroscopic data, runs the heavy computational models, and then sends a simple recommended set point back down to SCADA's control layer.
So SCADA is the muscle executing the command,
And PAT is the analytical brain figuring out what the command should be.
which is an elegant setup, but it leads us directly into a massive operational headache.
How so?
Well, you have SCADA running the physical machine, you have PAT acting as the analytical brain predicting the biology. How does a highly regulated pharmaceutical plant keep track of all this data flowing back and forth legally and efficiently?
Ah, yes. The compliance piece.
Right. It requires a third layer of orchestration. If SCADA is the commercial oven and the thermometer, just keeping the kitchen from burning down,
I love this analogy.
and PATA is the head chef actually pasting the soup and adjusting the spices based on the biological reality of the flavor.
Right.
The third layer, the MES and electronic batch record layer, is the health inspector and restaurant manager combined.
That's spot on. They aren't cooking and they aren't tasting. They are standing there ensuring every single step the oven and the chef took was perfectly documented,
Exactly.
ensuring that the raw ingredients were sourced from approved suppliers, and that the entire process complies with strict health codes before the plate ever leaves the kitchen.
Right. Manufacturing execution systems handle the workflow, the electronic signatures, and the batch genealogy. Because, legacy plants used to try and force SCADA to be the oven, the chef, and the inspector all at once.
It was an absolute nightmare for the operators.
Oh, I can only imagine.
It created massive bottlenecks and mountains of manual paperwork. Operators were literally walking around with clipboards, writing down values from a SCADA screen to type into a separate compliance system later.
Just begging for human error.
Exactly. By segregating these tasks into a specialized digital stack, you drastically reduce manual transcription. SCADA provides a trusted real-time infrastructure, PAT provides biological insight, and MES provides compliance orchestration.
And the data just flows seamlessly up the stack,
Seamlessly.
which means batch review and release, a process that used to take weeks of quality assurance teams manually verifying paper records becomes vastly simpler.
Because the system has automatically documented that the biology was controlled perfectly.
Right. And the sources we are looking at don't just stop at this current state of the art. They point heavily toward the cutting edge.
Here we go.
Here's where it gets really interesting. We're talking about how AI, sensor fusion, and cloud computing are making these three layers incredibly interoperable. Let's look at sensor fusion, where we are combining NIR and Raman spectroscopy into a single predictive model.
Well, fermentation broth is an incredibly hostile environment for optical sensors.
I bet.
It is a multi-phase chaotic system. You have liquid media, solid suspended cells, massive agitator blades churning the liquid, and millions of tiny gas bubbles constantly rising to the top.
Stuff a mess.
Yeah, a single pulse of near-infrared light might hit a microbubble, scatter unpredictably, and feed a false reading into your model.
It's just physical noise blinding the sensor.
Exactly. But by combining both near-infrared and Raman, that's sensor fusion, the system can cross-reference the physical interaction. The algorithms learn that bubbles distort NIR in one specific way, but distort Raman in a different way. So the AI can mathematically cancel out that physical interference, isolating the true chemical signal of the broth. It vastly improves the robustness of the measurement, especially when you scale up to massive 10,000-liter commercial reactors.
That is brilliant. The sources also detail how AI is developing transferable PLS calibration models for these sensors. Because historically, if you built a PAT model for one specific product in one specific tank, it was practically useless anywhere else.
Pretty much. The geometry of the tank or the specific lighting would throw the math off.
But now deep learning models are becoming sophisticated enough to isolate the biological signal from the reactor-specific noise.
Yes. You can train a model on a small benchtop bioreactor in a lab and mathematically transfer that calibration to a massive production facility without starting from scratch.
And on the data side, all of this complex PAT output, these AI-driven soft sensors, are now being pushed directly back into the SCADA historians.
Which is a huge workflow upgrade.
Right. So to an operator looking at their HMI screen, this impossibly complex, mathematically derived biological prediction just appears as another standard tag on their dashboard right next to the temperature reading.
It's totally seamless.
Furthermore, from that historian, the data is streamed directly into enterprise cloud platforms.
Where pharmaceutical companies can perform cross-site benchmarking.
Right. They can look at the metabolic trajectory of a fermentation batch in Singapore and compare it in real time to a batch running in Ireland.
Adjusting their global models dynamically based on ambient conditions or raw material variations.
Okay, I have to ask the hard question here.
Bring it on.
We are talking about AI-enhanced sensor fusion, running complex multidimensional math in the cloud and telling a model predictive controller to autonomously change the nutrient feed in a physical tank based on a virtual soft sensor.
Sounds futuristic.
It does. But how on earth do regulators or even the operators themselves trust an AI model to run a GMP, good manufacturing practice fermentation? It sounds like we are handing the keys to our medicine supply over to a black box.
Well, what's fascinating here is that the solution to the black box fear is exactly why the MES data integrity layer is so critical.
Oh, okay.
The AI is not operating in a vacuum because the architecture integrates those PAT tags seamlessly into the validated SCADA historian and ties that directly to the electronic batch record in the MES. Transparency is mathematically enforced.
So there's a paper trail, well, a digital trail.
Exactly. Every single prediction the AI makes, the confidence interval of that prediction, and every single action the controller takes based on it is permanently logged with an unalterable audit trail.
So if a batch deviates, you aren't just shrugging your shoulders at the AI. You can rewind the cape perfectly and see exactly what mathematical assumption triggered the physical change.
The MES layer takes the mathematical black box of AI and wraps it in a perfectly compliant, transparent glass box.
A glass box, I love that.
Yeah, and this supports a regulatory framework called Quality by Design, or QBD. Instead of manufacturing a drug blindly and just testing the final vial to hope it meets specifications, you prove that the process itself was continuously monitored and controlled within optimal biological parameters from start to finish.
Which enables the holy grail of biomanufacturing, real-time release.
Real-time release, yes.
The medicine is fundamentally approved the moment the batch is finished because the system has continuously proven its own quality every millisecond of the run.
It's incredible.
It really is. Let's bring this all together for you, the listener. If you are managing a bioprocess, scaling up a new biomaterial, or building a new facility, the practical takeaway is understanding the strict boundaries of your digital brain.
Right.
SCADA defined your data backbone and your control boundary. It is the autonomic nervous system.
PICE-POLIT defines how much of the actual biology you can see and anticipate. It is the analytical brain.
And the MES layer defines your compliance and workflow.
The health inspector.
Exactly. The facilities that will dominate the next decade of the bioeconomy are the ones making these three distinct layers seamlessly interoperate.
Because this technology stack is what enables humanity to scale up life-saving pharmaceuticals, create sustainable biomaterials, and transition away from fossil fuels safely and efficiently.
We are finally learning how to make the biology work for us at a massive scale by illuminating the black box.
It really is a paradigm shift in how we build the physical world.
It is.
Building on that shift, consider the long-term trajectory of this technology. We discussed how biomanufacturing relies on physical probes for the basic environment and soft sensors to translate light into biological insight.
Right.
But as AI-enhanced sensor fusion and digital twins become flawlessly accurate at predicting cellular behavior, we are approaching a fascinating threshold.
Look at that.
We could eventually see a future where physical probes for quality attributes become entirely obsolete.
Wait, really? A commercial fermenter running blind on the hardware side, but perfectly illuminated on the software side.
Exactly. We could reach a point where regulatory bodies approve a batch of life-saving medicine based entirely on the mathematical perfection of a virtual model, trusting the continuous calculation over the physical hardware.
Trusting the math completely over the machine. Now, that is a wild thought to leave on. Thank you for joining us on this deep dive. Keep questioning how the world around you is built, and we'll see you next time.
You've been listening to Biomanufacturing and Fermentation Technology with Prasad Ernala. Thank you for joining me. Until next time, keep engineering biology into reliable manufacturing.